Can you see that?
Yes, I can see it now.
All right, so dysautonomia, we just watched the brief introductory video. So, and first, what I want to cover is what is dysautonomia. It's a group of medical conditions and diseases caused by a malfunction of the autonomic nervous system. This image on the right shows some of the different syndromes and diseases term. So you have POTS, orthostatic intolerance, familial dysautonomia, multiple system atrophy, and then the ANS is responsible for involuntary functions, and that's like heartbeat, digestion, and breathing. When the ANS doesn't function correctly, it can cause heart and blood pressure issues, and then it can also cause difficulty breathing and loss of bladder control. It affects men and women equally, but some of these syndromes and diseases are more prevalent than women and men, and vice versa. It can range from mild to severe and even be fatal, but rarely. The only one that's regarded as extremely fatal is MSA, multiple system atrophy. And then there are primary and secondary forms of dysautonomia. Primary dysautonomia is when it occurs by itself, and then secondary dysautonomia is when it occurs in conjunction with another disease. So genetics of dysautonomia it's caused by different SNPs all over your genome, but most on chromosome 9. And then each SNP causes different symptoms and side effects. The SNPs are mostly on the IKBKAP gene, but they occur over all autosomes. So essentially, it is not a sex-linked disorder. It is also common on the ELP1 gene, and both of those genes are on chromosome 9. Dysautonomia is a recessive trait. You must have both copies of the mutations on the gene to have the disease. And each of these genes is IKBKAP is an inhibitor of capillite polypeptide gene enhancer. And then it's encodes for the IKAP protein, which is ubiquitously expressed at varying levels in all tissue types, and that includes brain cells. And then the ELP1 gene codes for the elongator complex protein, and that helps in cell motility, structure, and cell growth and development. And more depth on these two genes is the ELP1 gene produces the elongator complex protein, and then most individuals with dysautonomia both have mutated copies. There are some rare cases where they're heterozygous, so they have one copy of the mutated gene, but in all of those individuals observed, their symptoms are very, very mild. Dysautonomia disrupts how the protein is put together because there's different nucleotides, and that codes for different amino acids, which influences the protein. And that is important because with proteins, shape determines function. And if it's not in the correct shape, it can be targeted to be destroyed, or it cannot do what it's supposed to be doing. And it also reduces the amount of protein that is produced. Inconsistent mutation, some cells are unaffected and have the protein, and some are overly affected, which includes brain cells and have of the protein. In the IKP gene encodes for the protein called IKAP, and it's a scaffolding protein, and it is also involved in pro-inflammatory cytokine signal transduction, which is basically just a fancy way of naming something that plays a pivotal role in the immune response. And over here in the image on the right is chromosome 9, and the red line is where the ELP1 gene is located, and the blue is where the IKBKAP gene is located. So they are very close together. And here's an image of what happens with POTS and the IKAP protein. On the left is the normal protein production, and you can see that when they're making the copy and getting ready to perform, it excises introns 19 and 20, which is what are supposed to do. It's supposed to keep exons in and introns out. And then you wind up with the correct form of the protein, correct form of RNA, which then becomes a protein when they go into a ribosome, and then the tRNA comes and starts linking amino acids in a polypeptide chain. What happens in dysautonomia is exon 20 is cut out when they are making the protein or the mRNA strand. And what you wind up is exon 19, exon 21 when you have the mRNA strand, and you're missing this exon 20 here. And the protein doesn't function properly. And what happens is targeted for nonsense mediated decay, and then that have deficiency in your body. And as we said earlier, it is an inconsistent mutation, but one of the cells that are extremely affected are brain cells, neurons and glial cells included. just some statistics about it. Famous people with it are Nick Foles, Solange Knowles, and Ella Woodward. As of right now, there is no permanent cure, but precision medicines treatments are in the works. You can also take medication to alleviate the symptoms. Worldwide, over 70 million people are affected. I am one of them. And then specifically which version I have is POTS, which is a primary form of dysautonomia. And it's quite strange because I'm sure as you heard in the video, they said it occurs mostly in women around the age of 25, and I'm a male of 15, which is less than 2% of people who have POTS. Well, women around the age of 20 to 30 are around 80% of people that have POTS, so that was lucky. And then it normally takes years to be diagnosed, and that's due to lack of public awareness and professionals in the medical community. There are not many people that specialize in this. There's less than 10 doctors in Illinois that specialize in POTS. So multiple system atrophy is the most rare form of dysautonomia, and it has a 70% mortality rate, which is, it's good that it's the most rare because it is by far the most fatal. Dysautonomia is not fatal. Current medications and lifestyle choices, as well as neurotherapy, can lessen the symptoms of dysautonomia, but not cure it. So here are most of the symptoms for dysautonomia. Again, as you saw in the video, there are many, but here are pretty much the bigger ones and hallmarks of it. And the ones with the green dot next to it are the one I have. Symptoms include balance problems, noise and light sensitivity, shortness of breath, chest pain, discomfort, dizziness, swings in body and skin temperature, ongoing tiredness, visual disturbances, difficulty swallowing, nausea and vomiting, fast or slow heart rate, brain fog, large swings in heart rate and blood pressure, weakness, mood swings, fainting and a loss of consciousness, sweat less than normal or not at all, sleeping problems, migraines or frequent headaches, dehydration, and frequent urination. So, brain and cells on dysautonomia. Main regions of the brain affected by dysautonomia are frontal connections to the pontine nuclei have trouble forming, and these are the neurons that connect to the pons. And what the pons does, it coordinates balance in the head, neck, and body, and plays a primary role in eye movement, sleeping, dreaming, digestion, and swallowing, breathing, and heartbeat, which is where you get, tachycardia and the blood pressure regulation, which are hallmarks of dysautonomia. Also, the vagus nerve is weaker, or vagus nerve is weaker than in a normal brain. And what that does, it helps regulate your autonomics and has parasympathetic control of your GI system. So again, we're seeing that there's less control of your autonomic nervous system. And then the default mode network is hyperconnected, and the DFM, default mode, or DFN, default mode network, includes the mesial prefrontal cortex, posterior cingulate cortex, hippocampus, precuneus, and the inferior parietal lobe, as well as the temporal lobe. And what this causes is the patient tends to be unable to get into resting mode and contributes even more to the tachycardia effect. It also causes demyelination in the medulla and the pons, and also a loss of cells in the autonomic ganglia. And then specific cell types affected in dysautonomia are any cells which utilize the elongator complex protein and IKAP protein, which includes neurons for their cytoskeleton, dendrites, and axons, which they utilize the IKAP protein because it's a scaffolding one. They use it for cell structure. And then neurons and glial cells and cell motility. It's vital to get the cells where they need to be in the brain because that's how they form, the neuronal connections with the synapse. And then it also causes mast cells to have problems with activation. Neuroscience branches related to dysautonomia are epigenetics, and then Natalie's Share and Tell article accounted many factors of epigenetics that contributed to dysautonomia, and these include anxiety, diet, and culture. So there's an owl outside that's making a lot of noise. In neurogenetics, precision medicine can be helpful to fix various SNPs that cause dysautonomia, but these are still very, very basic treatments. There haven't been trial runs yet. They're so far mostly theoretical. But techniques like CRISPR-Cas9 could be used to this purpose, and also many others. And then lots of unknowns in these two subjects, and like how different environmental and physiological factors contribute to why a series of SNPs will give someone dysautonomia, but it will not give it to another person. And it's very similar in Alzheimer's disease to this matter, because a series of SNPs that gave someone Alzheimer's disease did not give someone another. Current hypotheses and ongoing research is main hypotheses are it's caused by the mutations on ELP1 gene and IKBKAP gene, caused by head trauma, and also by drugs that reduce cardiac output and preload that inhibit the ANS. All of these hypotheses they all have merit, and it's suggested that each of these do cause dysautonomia. Currently, the most important ongoing research is GWAS studies to see how dysautonomia can exist in correlation with another disease like diabetes or muscular sclerosis or Parkinson's. And there are studies to figure out a treatment to cure the underlying condition and not just the symptoms, which is what has to be done now. And landmark discoveries are figuring out a lot of SNP mutations occur on chromosome 9, and specifically the ELP1 and IKBKAP genes. And then also neurotherapy is successful in treating many patients with dysautonomia. For example, that helped me a lot. Future landmarks to hit are gene therapy and precision medicine treatments. It was moving around a few minutes ago. Okay, sorry, there's a bird stuck in the window right now. Dysautonomia, when it's a secondary condition, means it exists with another disease, and the highest chance of existing as secondary condition includes diabetes, Parkinson's, and muscular sclerosis. And this happens due to some shared SNPs between all the diseases, and because of the shared SNPs, it also shares many, many symptoms, particularly with Parkinson's, and this includes tremors, poor sleep patterns, rigid muscles, fatigue, and also dizziness. And dysautonomia related to COVID, it can be worsened from COVID infection, and specifically the POTS variant of dysautonomia is worse affected by COVID. Then vaccines are okay to get, and they are highly encouraged. They don't need to be altered for persons with dysautonomia. And then by physicians, it is recommended all persons with dysautonomia immediately get vaccines as soon as possible. But unfortunately, people with dysautonomia were not eligible for vaccine early use unless it was the MSA version of dysautonomia. And then because I have POTS, I took a special interest in presenting it here. And here are COVID-induced POTS mechanisms that you want to pay attention to are the ones that cause acute POTS and chronic POTS. And then what happens when COVID's viral entry, then you can have a hyper-responsive of immune system, and then it can lead to all these side effects, and then it leads to acute POTS, which is basically it's short-lasting. Any acute disease is not permanent, which chronic diseases are. And how COVID can influence chronic POTS is many ways, including non-immune mechanisms, downregulation of CD4 and CD8 T lymphocytes, transient immunodeficiency of both innate and acquired immunity, and then it goes along both of these pathways, and it will lead to chronic POTS in the...
Just a quick intro. I'm sorry, Ed, just to interrupt. Guys, the CD4 and CD8, that's helper T cells and cytotoxic T cells that we talked about. They just have these other names, just in case you were wondering. Oh, very good job so far, Ed.
Thank you. And then here we have more damage to nervous system and ANS, especially here. And once it leads to chronic POTS, you have POTS, essentially. There's no way to get rid of it. You can only alleviate the symptoms. So how I would go about treating dysautonomia. First one, without superpowers. So first I'd take a DNA sample of one cell, because obviously they contain all of our genetic code in each cell. And isolate the DNA by DNA extracting, and then we don't need to watch this video. This was just extra in case someone wanted to do it. This is an extracting DNA from a banana experiment. It's quite interesting and it does work. Third, I would do polymerase chain replication to make a lot of copies of the DNA, and then also use next-gen sequencing to sequence the DNA that results, and then compare that individual's DNA to a normal copy, and then discover the SNPs causing dysautonomia, and then find a viral deliverant mechanism that specializes in targeting where I want to cure. Then finally, you put CRISPR-Cas9 into a viral capsule with a strand of RNA telling it what to fix and how to fix it if it should come across the mutation. And then you inject it into the patient, and within about a week, it should show results in curing the disease because CRISPR-Cas9 works incredibly fast. Also, on the more fictional side of things, if I had superpowers to cure the disease, I would want the ability to be able to instantly know a person's genome and recognize, yes, the SNPs contributing to the disease. In this way, the precision medicine treatment could start now instead of having to wait for people to discover all the SNPs which cause dysautonomia, and it's different from person to person. And then once you discover the SNPs, you can immediately go ahead with the rest of this. We do not have to wait for genetic sequencing or even researchers now to discover what needs to be done, what's safe and what's not safe in treating patient dysautonomia with precision medicine. So a little blurb at the end here, living with dysautonomia. I would consider myself, a bit of an expert on that because I do have it. I have a more mild case of it. It's not terrible. I'm thankful it's not on the severe side. Most people's symptoms lessen as neuronal connections are stabilized and made in the ANS. It never officially goes away though. Unfortunately, I do have to take a lot of medication this to alleviate symptoms like low blood pressure and tachycardia, as well as inhibition of adrenaline receptors. And that helps with sympathetic and parasympathetic nervous systems because it causes the sympathetic nervous system to overfire and the parasympathetic nervous system to underfire. And by inhibiting the adrenaline receptors, it allows me to get into a resting mode, so to speak, so my heart's not always going insane. And then lifestyle choices to help alleviate symptoms of dysautonomia. This is more on the epigenetic side of things. Then that includes drinking lots of water, and then eating foods with higher sodium content helps. And I know that sounds counterintuitive, but what happens is water gets the body to produce more blood and fill your veins up so that they don't have to contract, 3 times, because that's quite difficult to maintain and do for patients with POTS. And if they're more full, they don't need to contract as much. And then the sodium also increases blood pressure, which helps, because there's high pressure in the veins, it's easier to move blood around and get to the organs it needs to, and all the other organs in the body as well. Avoiding hot temperatures also helps. And then avoiding standing up quickly because blood does immediately pull in the feet, and ANS doesn't go into overdrive to get the blood back up to the head, because by standing up slowly, you allow the ANS time to adjust to you standing up and is able to get all your autonomic functions under control. And then that's it.
Wow, that was a really awesome presentation. Wow. The amount of biology and background you talked a lot about. So I had a few notes that I really liked about your presentation as well that I'll just say off the bat. I liked how it was also personal, it was meaningful as well. You mentioned nonsense mediated decay, which is pretty advanced as well. I would say it's about, sometimes when we have these, when we have a gene that we have a messenger RNA, we have introns and we have exons. So a reason why 2% of our DNA, guys, just on the side is 2% of our DNA is protein coding. And there are a lot of other organisms, like even flies that we've looked at and worms, other organisms that have, I think, even more of their DNA codes for proteins. But ours is sophisticated. So we can have a gene, and a gene can encode multiple different types of proteins based on alternative splicing. So it's just about how exons are put together. When you have a messenger RNA, you are putting together different exons together, and you're splicing. It's just like how you're cutting and pasting different pieces of exons together. So that's a little bit more complicated, but it was really cool that you knew about nonsense mediated decay, how if you have a messenger RNA that is not going to make a viable protein, how there are other factors inside the cell that can get rid of it. So that's pretty advanced. I also liked how you talked, how we talked about how the 3D structure of proteins can give us functions, how you talked about the connection between COVID and dysautonomia as well. We were talking about the T cells, and we were talking about how if the body does not recognize self anymore, it doesn't know what is a self and what is foreign, then that can lead to issues, and how you talked about how you'd go about curing this condition. I know we didn't get to talk about CRISPR-Cas9, but I recommend you guys to check it out. These are modern techniques of gene editing that people are doing to increase the expression of certain genes in cells or decrease them. And I like how you also integrated the idea that we talked about SNPs, or people call them SNPs or SNPs. about one base pair change. And that's also, it's a really great way you said it about precision medicine, about just one DNA base being different. And we're trying to target, because in a lot of these complicated diseases, what these genome-wide studies that I'm telling you guys, like we're looking at, they're trying to help us identify, like, and they have these RSIDs, like where are there differences in that could give or increase the risk of developing certain conditions. So in my research, I'm trying to look at SNPs all across the DNA, even if they're not encoding regions, to try to identify if they can increase your risk of Alzheimer's disease. And my hope is, and my dream would be, if my my pipeline with Professor Wang can help people with all sorts of neurological conditions, and be a tool that researchers could use for their research in these conditions. So I really like that you took the idea of SNPs and talked about it and connected it with precision medicine. So I'm saying this as well for you guys, so that those of you who are working on your project, you could take these into consideration as well. I think you guys are doing a great job, and you did a really good job explaining all of the concepts about dysautonomia. So really good job, and thank you for going first again. Does anybody have any questions for Ed?